Hey class, today's video is video number 3 in this unit of meiosis, genetics, and heredity. Part 3 is going to look at Punnett squares and predicting possible outcomes of dihybrid crosses, and look at another model of inheritance, which we'll call a pedigree. So, to begin with, we have a dihybrid cross definition. What is it? It's really a cross involving two different traits. So we have two traits instead of just one. Before we just looked at flower color of pea plants. Now we can look at flower color and shape of the pea all in one cross. So in this case, we're going to use two different traits represented by one letter like we were before, only now there's two of them. So trait 1 is color, just exactly like before. P is purple, and then we also have the recessive white. All right. Shape, we're going to use R for our letter here, and R is round. The big R, the dominant trait of a P, is round, and the little r recessive trait is wrinkled. Parent 1 is going to be heterozygous purple and heterozygous round. So you can see their genotypes are both heterozygous right there. Parent 2 is going to be homozygous white and homozygous wrinkled. Since being white flowered and being a wrinkled pea-shaped are both recessive traits, they get the small alleles there. So you'll see how we represent this in a Punnett square. So two traits from each parent. Parents that we talked about on the previous slide are listed. The only thing that I need to put down here really is to note that this is the cross. What we do is we combine both traits together. So here we have our purple flower that's heterozygous, and here we have our round pea that is heterozygous, and we just stick them together. What we need to do to set up our squares, we have a 4x4 square instead of 2x2. So we're going to have 4 by 4, and we're going to have to put in the allele combinations that can exist when they're combined together. So the key here is going to be to FOIL. All right. So I'm going to go ahead and write that. This is going to be the key. So we say FOIL just like in math. You may have learned this before. The F is first. The O is outside. The I is inside. And the L is last. All right. First, outside, inside, last. That's the order we need to do this to separate our allele combinations for each parent. All right. So we'll switch colors here to red. Let's do this parent first. Okay. So this will be the one that ends up going up on the top. This will be the first one we do. In order to FOIL this, we're going to write out our different alleles. All right, and we need to do FOIL. So I'll try to use different color for each one of these and see if we can highlight this. The first is the first in each combination. So I'm going to go ahead and do red for that one. So the first of each set. So the first is this big P here and big R here. All right, we're going to combine those two and put them together. So here you go. There's big P and big R already in there for you. Okay. Now we'll switch to the next color. It's going to be purple. Purple is going to be our outsides, and then we're going to do those two combinations. So the outsides are going to be these ones, big P and little r. So you see that there, big P, little r. All right. And we can switch over to white to do the insides. And the insides, we're going to go ahead and have these two inside alleles together. So we have little p and big R. We'll go ahead and write it just like that. Little p and a capital R. And then the last one, go ahead and use green for last. All right, as the last of each pair. So it's this little p and little r right there. All right, so that's one parent. That's how we set up our dihybrid cross up at the top. And then we have to do the other parent, who will do the same order. I'll start them in red, And this parent here is a little easier. Those of you that can look at probability of things right now, and you start to identify, well, hang on a second. If he's recessive for both, then really, if we FOIL this whole thing, is there any combination that they could have that isn't a little p and a little r? And we'll go through the process again, but you're going to see that that's where all of these come from. All right. So go ahead and do the firsts. The first of each pair right there. Little p, little r, and then we go ahead and move to our purple, and then that's the outsides. So here's the little p and the little r there. So that's the second one. Then we go to white and do the insides, little p and little r. And then we can go up back to green and do the lasts of each one, which is again a little p and a little r. All right, so no matter how you slice and dice it, they're going to get both recessive alleles distributed there. So that's where we get all those from. Once we have the parents set up, then we're doing the same thing we did in our monohybrid crosses, except for we have two traits that we're bringing together. So in this case, what we're going to do is still drop those letters down, but we're going to put them back together. So we're putting the P's together and the R's together. We're still going to list the dominant allele first when we put this all in place. So for this one, we're going to take this big P and that little p and stick them back together. Big P and little p. And then we're going to take our big R and little r and stick them together. Just organize it like that. It makes a little more sense when you go through. If we do this all the way down, you're going to see it's all the same thing, because this combination mixed with this is going to be the same all the way down. Same here. We see that big P, little p, and then little r and a little r. And then we go all the way down. Now this H is here by mistake, so let's go ahead and write right over top of that. Big P, and little r, and then the same thing all the way down. And then we keep going here. So we have a little p and a little p. So our little p go together. The big R goes first and the little r goes next. We go ahead, and that's going to be the same all the way down. Now, not all of these dihybrids are going to be the same for each column like these are. This just is because it is a unique, because all of these alleles are only the same. So they're homozygous recessive for both traits. That's why each column is identical. And the last column here, a little p and a little p and a little r and a little r. So we got all that. And again, this is going to be the same all the way down. All right, so there's our dihybrid cross. Now what we need to do is follow two traits, and this becomes a little more complex when we start talking about probability and different percents of the traits that are there. Instead of just saying which ones are purple or which ones are white, now we can say which ones are purple and wrinkled, which ones are purple and round, which ones are white and round, which ones are white and wrinkled, and it adds a whole nother level. The final thing we need to talk about real quickly here is a pedigree. Pedigree is a pretty simple graphic representation of genetic inheritance. You really just need to know what the symbols are. The symbols represent different traits throughout generations. So I'm going to mark this up just a little bit here so we can see what we're talking about. Males are square and circles are females. So we can go ahead and identify those pretty easily. Anything that's dark and shaded is an affected individual. They either have a trait or a genetic disorder, something like that. Generations are usually numbered in Roman numeral. So in this, we have 1, this is generation 2, generation 3, and 4, all the way down here. And that's in each kind of level, all right? And then individuals are usually numbered. So we number this like individual 1, individual 2, and so on all the way through. All right. Anything special that you see, if they're connected by a line, that means there was mating, they had children. Offspring are in birth order as well, so we list them like left to right in their birth order. And twins, if we ever have that, can be represented with a separate crossing here. Non-identical twins, same thing. They come from one parent at the same time, so they split off on an angle like that. And identical twins, like we said, are connected. The only other thing we need to talk about is what is a carrier. A carrier is somebody that has a recessive allele for the trait, or has an allele but it's not shown. So it has the allele for a trait that we're following here. Carriers can be represented by half-shaded individuals. So for example, we could have this guy here in generation 2. We can mark up our pedigree this way if we know that this guy's a carrier for a recessive allele. So what does that make him? He is probably heterozygous. All right, and that's what a carrier is, probably someone that's going to be heterozygous for a specific trait.